Steve Reichow, Assistant Professor of Chemistry in the College of Liberal Arts and Sciences. I guess what we describe ourselves as are molecular photographers. So we study biology, and we're interested in the aspects of biology that are too small to see with the human eye. And so we use really high-powered microscopes to allow us to peer into the molecular world that goes on inside of our bodies. This is a field of research that's interested in understanding the three-dimensional structures of the molecules that live inside of our cells, mostly proteins or nucleic acids. And at the time I was being trained in a technology known as NMR spectroscopy, which has some inherent limitations about the complexity of the types of molecules that you can study. And so when I was looking towards my postdoc, an emerging field of structural biology known as electron cryo microscopy, or cryo-EM, was being developed and becoming more and more popular. And so I entered into that field, and this is where you're really just taking direct pictures of the molecules you want to study. And there's really no limitation of the type of molecule you can characterize with this technique. And I just fell in love with it, and so this is the technology that I brought with me here to PSU. One of the major projects that we're investigating in my laboratory has to deal with trying to understand how the cells in our body communicate with one another. And so one of the most direct ways of doing this is through a class of proteins known as the gap junctions. These are proteins that form a direct pathway for cells to communicate with one another, to pass chemical information back and forth. And so we're using electron cryo microscopy to visualize these channels and understand how they open and close and facilitate this kind of network of chemical information flow in the body. My name is Steve Reichow, and we're using electron cryo microscopy to visualize the molecules of life.